Thanks everybody for coming. Today I'm super excited to invite my friend and colleague today to talk about disarming immunity, specifically about understanding how Zika virus subverts cell-mediated immunity. That's how these little boogers get through. And this is Aaron Carlin. He's an associate professor in pathology and medicine at UCSD, and he is associate director for our clinical micro lab. And he's also the assistant director from our MSTP program, where he is a graduate. And he's also the director of our emerging virus BSL-3 research program. So lots of hats, lots of connected hats. Really happy to have him come and talk about his work. Next slide. So he runs an independent research laboratory at UCSD looking at emerging and re-emerging infectious diseases and their interactions with the immune system. As I said, he's a graduate of our MSTP program, and his thesis was on group B strep and how it disarms the innate immunity utilizing molecular mimicry and direct protein interactions to engage inhibitory sialic acid binding immunoglobulin lectins, Siglix. Anyway, that leads to exactly what we're going to be talking to about, well, similar to what we're going to be talking about today in terms of rather than strep, it's going to be Zika and its evasion of immune cells. Also at UCSD, he was in the PSDP program, the physician scientist training pathway, where he did internal medicine residency and ID fellowship. That's where we got to know each other. And he also did a fellowship in Chris Glass's lab, developing unbiased genome-wide approaches to look at host-viral interactions. And he's received several honors, including funding through the BWF Career Award, NIAID K08, who's got an ASCII Young Physician Scientist Award. Lots of good stuff. Next slide.
All right, that's actually it for the introduction. One other thing I want to note to the audience, though, Dr. Carlin is actually one of the first recipient of our Prepare Developmental Grant Awards, and that's what his talk today is related to. So whenever you're ready, Dr. Carlin, go ahead and take over.
All right. All right. Can everyone see my screen?
Yeah, looks good.
All right. Thanks for joining today. I'm really excited to talk about some of our stories related to Zika virus and how it interacts with our immune system and how it really subverts those innate immune cells early in infection to drive infection. So this is a brief outline of kind of how we'll approach today's talk. First of all, I'll tell you about Zika virus and why I think it's really important to understand this virus and really continue to think about it despite its absence in the news over the last 3 or 4 years. I'm going to tell you about our integrated genomics method that we've developed that allows us to discover virus-regulated gene programs in infected cells and why we think that that's a very cool method and important for understanding virus-host interactions. And then I'll tell you 3 stories based on that method where we look at effects on type 1 interferon signaling, lipid biosynthesis, and then pro-inflammatory gene signaling. So Zika virus is in the family of flaviviruses. There are many important human pathogens in this group. Zika is down here, closely related to dengue virus and also yellow fever virus. And then also viruses like West Nile and Japanese encephalitis virus are in this group. So many of these viruses are really important causes of human disease. Zika virus is primarily transmitted by mosquitoes, Aedes mosquitoes, like Aedes aegypti. And Aedes aegypti live in tropical and subtropical parts of the world. And they really have adapted well to replicate in urban environments and oftentimes cohabitate with humans, making them a really important vector for the transmission of human disease. If you look at where Aedes aegypti mosquitoes are, this would basically about 1/3 of the human population are at risk of infection or bites with these mosquitoes, and therefore a huge portion of the population is at risk for transmission of Zika or dengue into these populations. And we think that a large portion of this population is not already infected with Zika and therefore would be at risk of kind of new infection with Zika. So a huge portion of the population is still at risk for infection. Zika virus, although it's transmitted by mosquitoes, really its most important clinical consequences is when pregnant women are infected with the virus. The virus can basically cross the placenta and infect the brain of fetuses in utero, vertical transmission. And that can lead to either loss of the fetus in about 5 to 10%, we estimate, of infections, or the development of, many complications, including devastating neurologic complications like microcephaly. The rates of that vary widely from something about 5% to about 40%. So we don't know the exact numbers. And we've also found that infants that we thought were normal at birth actually went on to develop neurologic sequelae later on in development. And so this is an incredibly devastating disease when neonates are infected. In contrast, in most adults who are infected, the disease is almost silent. About 80% of individuals never know they're infected, and those that do know they're infected have very mild symptoms generally, and many don't present clinically to either the doctor or the hospital. And so we basically have a disease that can be transmitted basically silently from individual to individual, but can have devastating consequences in pregnant women. And really, a huge portion of our population is still at risk of this transmission. And despite the fact that Zika virus has not been in the news since really the American outbreak in 2015-16, we still see small outbreaks of Zika virus in places like India in 2022 that show us that the virus is still circulating. And since it can circulate almost silently, it will be very hard for us to pick up these continual outbreaks that occur. So it's very important to understand this disease and how we can potentially prevent the complications associated with it. Today, I'll also be talking about dengue virus disease, kind of as a way for us to understand how Zika virus is different from other flaviviruses. Dengue virus is also transmitted by Aedes mosquitoes. It causes about 390 million infections per year and kills a person every 12 minutes. Its clinical manifestations are very different from Zika virus. There can be asymptomatic infections of dengue, but we think that it's not as common as Zika virus infections. And those that are infected and symptomatic have, much more severe clinical symptoms. They have, severe headache, muscle pain, joint aches. And so the clinical manifestations are much more inflammatory than in typical adults infected with Zika virus. Interestingly, dengue virus typically kills people through the development of shock and multi-organ system dysfunction. And really that shock occurs after peak viremia. So the virus peaks, and as it's falling due to the introduction and increase in adaptive immune responses like IgM and T cell responses, it's during that fall and viremia and increases in adaptive immunity that you see the risk of shock develop in these individuals. And so understanding how the immune system can lead to this or cause or contribute to shock is also a really important question that we're interested in answering. And so Zika and dengue have very different clinical manifestations, but genetically and in terms of life cycle, they're very related. So Zika and dengue share about 55 to 60% sequence similarity. For comparison's sake, dengue virus is broken into 4 types, 1, 2, 3, and 4. And each of those types only share about 60 to 70% sequence identity. And so Zika virus is highly related to dengue virus. Both viruses produce a 5' capped positive sense single-stranded RNA with basically no polyadenylation secondary structure at the end to terminate and prevent degradation. That capped RNA basically acts as a messenger RNA and can be directly translated into a polyprotein that is then cleaved by proteases to form 10 proteins, 3 structural and 7 non-structural proteins. The virus, after it's entered and made its proteins, causes invaginations in the endoplasmic reticulum where it replicates. And then the virus buds into the ER and then out through the Golgi, and then basically is released extracellularly. And so both dengue and Zika, share a lot of these similar traits. Similarly, both Zika and dengue infect myeloid cells. They're kind of the primary early target of infection, and they are responsible or play a really critical role in the dissemination and propagation of the virus. So these are the cells that really should be protecting us from viral infections, and instead they're amplifying virus. And so infected DCs, resident DCs are infected early in infection. Those infected DCs propagate virus, are thought to make cytokines that recruit other innate immune cells into this area, which are then infected. And these and can also move back into the lymph nodes and lymphatics to drive systemic spread of the virus. At later time points, it's thought that then the innate immune system or the adaptive immune system kicks in and then starts destroying infected cells and allows for kind of control of viremia later in disease. So in this case, your innate immune system is a primary target of infection and basically helps to propagate the virus. Given that Zika virus and dengue virus had such different clinical manifestations in humans, we made a hypothesis that potentially differences in the way that our innate immune system responds to these viruses early in disease may dictate or may help explain, at least partially, why the systemic manifestations of these diseases, these infections are so different. And so we went about trying to understand how Zika virus and dengue virus, manipulate or replicate in dendritic cells. And so one important thing that I wanted to discuss is the system that we use to do this, which I think is really critical for understanding why we're able to kind of understand this virus at this level. So when these viruses, when we have models looking at virus infection in vitro using primary human cells, generally the majority of cells are not infected. Most cells are able to basically restrict infection and prevent infection, whereas highly susceptible cell lines, oftentimes with enough virus, you can infect almost the entire population. But in these primary models, a vast number of these cells are uninfected. And so if you take a system where you look at the response in this population, you have a mixed response. You have things that are occurring in uninfected cells and things that are occurring in infected cells. And this obscures your ability to really understand how the virus is controlling the cell. And so we developed a method where we can actually fix these infected and uninfected cells, these primary cells, and then basically use FACS to separate them into infected and bystander populations. And this is really helpful for us to be able to separate out what's happening in the infected cells. Additionally, it gives us a really excellent control by which we can compare what's happening in the infected cell. The bystander cells are not infected, but they should be responding to all the environmental signals that are present in the same dish that are with the infected cells. And so if a cytokine is released or a growth factor or something else, they'll respond to that signal. And we should see the same response in the infected cell. And so using this method, we have developed the ability to do many types of genome-wide approaches, including transcriptomics and many genomic methods. And then we're able to compare these bioinformatically to identify, basically patterns that are missing in infected cells that are present in bystanders. So this would tell us that this is likely a suppressed pathway in the infected cell, and pathways that are upregulated only in virally infected cells, but not in bystanders. Really identify those pathways that the virus is specifically upregulating. And then therefore we can identify these pathways and we can investigate whether these pathways are truly proviral and things that are being upregulated by the virus, or antiviral that are being suppressed, and understand that in a deeper way. So using this method, we took human blood and monocyte-derived dendritic cells and infected them with Zika or dengue virus, and went through this protocol of formaldehyde fixation, intercellular staining, and its separation, followed by total RNA sequencing. And as you can see, this nicely separates cells into infected populations. We get a lot of RNA from the virus in the infected populations, but very little to no RNA in the uninfected populations. And you can see how good these viruses are at replicating. By 24 hours post-infection, almost 20% of all the RNA reads coming from inside of an infected cell are from the virus. So the virus is incredibly efficient at replicating. Interestingly, when we did PCA analysis to look at responses in the infected and bystander cells, we quickly noted two things. So one, that the infected cells respond very differently than the uninfected cells, the bystander cells. We hypothesized this and we thought this was a good reason to separate these. But we also noted that the response to Zika and dengue is very different. And so we were very interested in trying to understand that at a deeper level. And so when you look at this, you clearly can identify genes that are regulated in either an infection and or virus-dependent manner. So in this case, this receptor, which is a receptor for trail that induces apoptosis, is only upregulated in infected cells, Zika virus positive or dengue virus positive cells, but not in the bystanders. In contrast, IFIDM3, a typical interferon-stimulated gene, is only activated in bystander cells in both groups, but not in the infected cells. Whereas SCD, which is involved in fatty acid biosynthesis, is only upregulated in both a virus-specific and infection-specific manner, only in Zika virus positive cells. And TRIB3 is upregulated in, again, a virus-specific, infection-specific manner, so only in dengue virus, and TRIB3 is involved in ER stress. And so this allows us to start understanding how how cells are responding to these viruses and how these viruses may be manipulating those signals. For the rest of the talk, please note these colors. Dengue virus negative cells will always be in this teal color, dengue positive, gold, Zika virus negative, blue, and Zika virus positive, red. For those of you in the audience who are colorblind, I try to choose these colors carefully so that they would be you'd be able to discriminate them. And so hopefully that is true. And please let me know if that's not the case after the talk, and I'll work on it. So as for the transcriptional responses, looking at, kind of a general pathway analysis, the first thing, if we look at virus upregulated genes and virus downregulated genes, what we see very quickly is that the viruses do not share overlapping transcriptional responses. These responses are largely virus specific. So all of these viruses are highly related. They upregulate and downregulate completely different gene programs. The dengue virus program in general is highly inflammatory, and the Zika virus program upregulation is really restricted to lipid metabolism, which we'll talk about later. And Zika virus strongly downregulates many of the genes that are critical for DC function. And again, we'll talk about that later specifically. So the first part of our talk is about type 1 interferons. And so what we see here is we're looking at interferon beta production on the left and interferon stimulated genes on the right. So type 1 interferon is made in response to viral infection. It's secreted into the extracellular environment and binds the interferon, type 1 interferon receptor. And that leads to JAK-STAT signaling and the production of interferon stimulated genes. And what you can see here is that virus infected cells in red and gold make more interferon beta than the bystander cells. So it's predominantly produced in infected cells. The infected cells themselves are able to make type 1 interferon. However, they cannot respond to type 1 interferon in either case, Zika or dengue. No interferon stimulated genes are produced. If you look at the kinetics of that, This is looking at the percent infection by antigen staining of virus infected cells. That between 10 and 12 hours, you start to see this big spike in the amount of infected cell population, which increases to about 18 hours and then levels off. This is a period of very rapid virus production. And if you're looking at viral RNA, you can see that here. Also, early production of viral RNA, really rapid production of virus. However, the interferon beta response, the production, that recognition of viral infection and production lags significantly behind that. So it's not really until you're out here at 18 hours and 24 hours and then really late that you start seeing massive productions of type 1 interferon, suggesting that the virus in these infected cells is replicating earlier than the production of type 1 interferon, and really that recognition of the viral infection itself is delayed to only the time point at which the virus is present. And really this is critical. If you treat these cells early with interferon exogenously at 6 hours, 4 hours post-infection, you can block infection completely. And so the fact that the virus is able to delay this response until late allows the virus to propagate in the infected DCs. And so that's very quickly the first part of this talk, that basically Zika virus stimulates interferon beta expression, but suppresses ISGs in infected cells. So Zika virus and dengue virus both do this. They both inhibit the type 1 interferon response, but not the production of interferon beta. Type 1 interferon is primarily produced in infected cells, so it is recognition of the virus in those infected cells, but it's late recognition that drives interferon beta. And that opens a critical window for replication. And really, if you don't separate the virus and the bystanders in order to look at these responses, what you would have concluded from this is that infected cells make interferon beta and interferon stimulated genes, but in fact, they don't. It's only by separating the infected from the bystander cells that you're able to understand that the interferon stimulated gene response is lost. And that's consistent with previous publications from Adolfo Garcia Sastre's group and others, that Zika virus is able to degrade STAT2, and dengue virus can degrade STAT1 and STAT2, and that allows for the blockage of the interferon stimulated gene production. Our second part of our talk is really going to focus on lipid biosynthesis. So if you look at what happens in Zika virus infected cells.
Aaron, can I ask a question?
Oh, yeah.
This is
Yeah.
Doug. So this delay in response, is that really different from other negative strand RNA viruses, because the cell has to recognize the RNA and then synthesize a new protein? I'm just curious if this is distinct or not.
I think it's actually really common for a lot of viruses. I believe that's been shown by quite a few other groups for influenza and some other negative sense-stranded RNA viruses. I think that it's not, if you think about pattern recognition, I think generally many people think of pattern recognition as initial recognition of the virus or the pattern on entry. And I think that that's not really true most of the time, that there's very little pattern to recognize early in infection. And it's only after massive production of pattern late that you get stimulation of these responses. And so I think it's in keeping with what I would expect, but is not always commonly thought of, I think.
Right, because I always thought of the interferon benefits to these viruses was for protection of the uninfected cells.
Yep. And that's absolutely what we see in this system, is that basically, although the initial cells can be infected and make virus, we see no secondary infection, because these are primary human DCs. They're highly responsive to interferon, and they basically create an antiviral state that does not allow a second round of infection. So we see no spread of infection. For the lipid biosynthesis, if you look at what's upregulated in Zika, really the upregulated genes are almost entirely restricted to genes involved in steroid, fatty acid, and cholesterol biosynthesis, which is quite remarkable and very different from dengue virus, where we see really no upregulation of these gene programs. If we look a little bit deeper and we look at what's happening in Zika virus negative versus Zika virus positive, the infected cells versus the bystanders, really this upregulation is only occurring in the infected cells, and that's comparing the infected to the bystanders. And that's true for both cholesterol metabolism genes and fatty acid metabolism genes. And you can see a few of them here. These are two genes involved in fatty acid biosynthesis, FASN and SCD, that really you only see upregulation in the infected cell population. And that's again true for genes like DHCR7 and HMG-CoA reductase. These are basically genes involved in cholesterol biosynthesis. And if you fact separate them like we do, you clearly see that this is true. Zika virus positive upregulation, significant upregulation. However, if you run the same protocol and don't separate the cells and just go through sorting, but just take the entire population, what you end up seeing is that you see no regulation of these genes. So this is a case in which sorting and separating the infected from the uninfected cells is critically important to being able to identify this regulatory program. Otherwise, it would have been lost because many of the negative cells are not upregulating this program. And so this again, shows the importance of our approach. Now, RNA-seq and transcriptional analysis is really great at telling us what happened in a cell. And telling us how a cell is responding. That RNA is likely to mate, not likely, but not always going to be made into protein. And so we can understand kind of what is happening in a cell, but it doesn't generally, it's not a great way to understand why that regulation happened. What were the transcriptional patterns, transcription factors that drove that response? And so we wanted to employ a method that would allow us to understand why lipid biosynthesis genes were being upregulated in Zika virus infected cell. And a couple of genomic methods that people have used to look at this include things like DNase hypersensitivity, or ATAC-seq would be a similar method, where you're looking at open regions of chromatin. There really gives you a nice, really focal peak, oftentimes over transcription factor binding sites. So really useful in identifying transcription factors. This is really great if you're looking at different types of cells. However, at least in my experience, the changes in DNase hypersensitivity or ATAC-seq is not very dynamic in a single cell type in response to a stimulus. So in a cell, in a DC that then is infected, we don't see huge changes in DNase or ATAC-seq peaks. And also the change in the peak doesn't always tell you whether there's actually active promoter or active enhancer underneath that. It just tells you about kind of the chromatin landscape. And so there are positive things about DNase or ATAC-seq, but some negatives. H3K27 acetyl is a histone mark that's often associated with active promoters and enhancers. And so in this case, it's great because it's telling you about the change in activity of an enhancer or promoter, but the peaks are really broad and there's tons of transcription factor binding sites underneath these peaks. And so it's very difficult to discern which transcription factor is really, mediating that change. So we used a technique that was developed here at UCSD by one of our collaborators, Chris Benner and Sasha Dudka, and it's called capped small RNA-seq. And I'll tell you a little bit about it and how that helps us to identify these regulators. So csRNA-seq is a method where we cut out or identify or grab very small RNAs, 60 to 20 nucleotides in size, and then we degrade all non-capped RNA, so you get rid of microRNAs. And really what you're doing is you're enriching for 5' capped short RNAs, which are basically newly transcribed RNA Pol2 dependent transcriptional products. And so what this does is it captures new or kind of nascent RNA synthesis or nascent-like RNA synthesis at both promoters, but also enhancers, because enhancers make eRNAs. And so basically, this tells you both, so csRNA-seq gives you a very sharp, nice peak directly adjacent to where transcription factors may be binding, to identify transcription factors, and is incredibly dynamic to allow you to identify activated regulatory regions. And so this technique is really powerful for identifying transcription factors that may be driving changes in activity. And so we utilize this in our basically the same approach that we used previously of sorting cells, fixing them, separating, and looked at the areas where csRNA-seq peaks were increased in Zika virus positive cells. And the top category was activation of gene expression by SRABPs, which are basically a family of our 2 transcription factors that are key regulators of lipid biosynthesis. Additionally, when we looked at those motifs that were upregulated in the Zika virus infected cells, we found an E-box motif, which can be bound by SRABP, as well as a CCAT motif, which is bound by NFY, a transcription factor known to collaborate with SRABPs to drive lipid activation. And so really what we were seeing is this production of SRABPs, and then SRABP moving to sites where lipid genes were. So to confirm that, we did use our same genomics integrated method, and we did ChIP-seq for SRABPs. And so what we found is that really SRABP is recruited to the genome really only specifically in Zika virus positive cells. And it's recruited to many sites of lipid genes, including the FASN gene here. This is the promoter. And you can see increased SRABP binding at 2 different binding sites where SRABPs are, the SRE motif is found. You see accompanied upregulation of csRNA-seq, so increased nascent transcription immediately adjacent to those binding sites, and then increased production of the SCD or the FASN gene and only in Zika virus positive cells. So again, correlating this increased recruitment of SRABPs to increase nascent transcription to increase production of the gene. So basically that showed us that SRABPs were likely responsible for this lipid production, but was this important for viral infection? And so we took advantage of the fact that normally during normal cellular response, cholesterol or desmosterol can basically feed back from the production of lipids to inhibit the activation of SRABPs. And so we took advantage of that, and we used a synthetic mimetic of desmosterol called DMHCA that can similarly trap SRABPs in the ER and block SRABP activation. And we found that the addition of SRABP decreased intracellular viral RNA by about 10-fold, but it decreased the release of infectious virions by between 1,000 and 10,000-fold in infected DCs. And so really potently inhibited the production and release of infectious virions. Furthermore, using add-back experiments, we found that the addition of cholesterol back to DMHCA-treated DCs could rescue that phenotype, suggesting that DMHCA was inhibiting Zika virus by blocking the SRABP's ability to make cholesterol. And so that is kind of the take home from this part of the talk. So, and specifically, we later tied this to SRBP2, which is the main controller of cholesterol biosynthesis. So we found that Zika virus activates lipid gene expression in a virus and infection-specific manner, right? So very different from dengue and only in infected cells. It stimulates SRBP activation and recruitment to lipid genes and associated transcription. This is pharmacologically targetable if this could be dispensed and block infection. And SRBP2 does this by increasing cholesterol production, which allows the increased production and release of infectious Zika virus. So again, interferon beta is made but delayed, and that allows virus to replicate. And the production of lipids allows the production of lots and lots of new virions to spread infection. So for the third part of our talk, we're going to focus on not what Zika virus is upregulating, but what it's downregulating. So if we look at dengue virus upregulation in DCs, we see cytokine production, we see leukocyte migration, chemotaxis, and upregulation of a lot of inflammatory gene expression programs. So this is kind of what you would expect a DC to do if it was infected with the virus. In contrast, Zika virus does not upregulate any of those genes, but it actually downregulates genes involved in antigen presentation, proliferation and differentiation, migration. So really the core functional responses that you would expect the DC to do, Zika virus is really downregulating these gene expression programs. So really suppressing the functional response of a DC. And so if you look a bit closer at cytokines, which are in purple, or genes involved in co-stimulation in pink, all of these genes are really expressed in a much higher levels in dengue virus positive infected DCs than they are in Zika virus positive DCs. So all of these are significantly upregulated. Additionally, if you look morphologically at the infected cells, Zika virus infected DCs look like they're still immature. They look like mock treated DCs, whereas dengue virus DCs have these kind of extended ramifications. They look very much like DCs that are treated with LPS, that are stimulated and activated and basically morphologically change into mature DCs. That RNA expression is also correlated with the production of actual and release of cytokines. So Zika virus infected DCs release very little IL-6 and TNF compared to dengue virus infected DCs. And this is true across Zika virus strains. So Zika virus Dakar is an African lineage Zika virus. FSS is an Asian lineage Zika virus that was pre-pandemic or pre-epidemic. And SD001 is an Asian lineage Zika virus that is from the American epidemic in 2015-16. And so this is true for all Zika virus strains across these very diverse Zika virus lineages. Similarly, if we look at cell surface expression of co-stimulatory molecules, CD40 and CD80, we see strong upregulation of this cell surface expression in dengue virus infected DCs, but not in Zika virus infected DCs. So basically no upregulation of the molecules that are really critical for T cell co-stimulation. And so when DCs need to stimulate T cells, they really send multiple signals that are integrated together to stimulate the activation and proliferation and polarization of T cells. That includes antigen presentation and MHC to the T cell receptor. Signal 2, co-stimulatory molecules on the cell surface have to engage to activate the T cell. And then cytokines are secreted, which help with activation and polarization of T cells. And so our data up to now would suggest that signals 2 and 3, at the very least, would be missing or inhibit or significantly less in Zika virus infected cells than in dengue virus infected cells. And so to see whether Zika virus infection inhibits T cell activation, we did a mixed leukocyte reaction shown here where we prepare immature DCs. We do virus infection for 48 hours, and then we do mixed leukocyte reactions for either 1 day or 3 days and allogeneic PBMCs. And we measure cell activation marks like CD69 or the production of cytokines through ELISA or intracellular cytokine assays. As you can see here for CD4 positive T cells, dengue virus stimulates significant CD69 production on these T cells, whereas Zika virus does not. You see the production of interferon gamma by these cells. You see massive amounts of interferon gamma production by dengue virus infected DCs when they're co-incubated with CD4 positive T cell or with mixed leukocyte reaction. And you see in a co-stain of CD69 positive interferon gamma for intercellular cytokine staining that the T cells themselves are positive for interferon gamma, showing that they're the cell type that's predominantly or primarily probably making this interferon gamma response, or at least involved in that interferon gamma response. For CD8 T cells, we see very similar reactions. We see less CD69 on Zika virus infected DCs, incubation with Zika virus infected DCs. We see much less CD69 positive, interferon gamma positive, CD8 positive T cells, and we see significantly less granzyme B marker of cytotoxicity activity and CD69 positive cells in the CD8 population. So again, the conclusions here is really that Zika virus infected DCs really poorly stimulate both CD4 and CD8 positive T cell responses, very, significantly less active than dengue virus infection. To try to understand why this was occurring, we compared dengue virus and Zika virus infected DCs and looked at the gene expression programs. And as you saw before, dengue virus strongly upregulated cytokine inflammatory responses, where Zika virus was predominantly lipid biosynthesis. And if you looked in the promoters of a lot of these genes that were upregulated, these 1,031 genes, there was a strong enrichment for NF-kappa B P65 motifs, which suggested that a lack of P65 activation may be important for the loss of this response in Zika. To confirm that, we did NF-kappa B P65 ChIP-seq using our integrated platform, and found that dengue virus infected DCs strongly upregulate P65 binding throughout the genome at many locations, whereas that response is very muted in Zika virus positive cells. And if you look at those 1,031 genes that are strongly upregulated in dengue positive versus Zika virus positive, 70% of those genes have increased P65 binding in dengue virus positive cells compared to Zika virus positive, again suggesting that a majority of this transcriptional program that's different is regulated or at least enhanced by the binding of P65. And you can see that for critical genes like IL-12, which is really critical for the stimulation of Th1 responses by DCs, you see increased P65 binding at both the promoter and an enhancer in dengue virus infected cells. And that corresponds nicely to increased synthesis of IL-12A that you don't see in any of the other populations. Next, we wanted to know whether Zika virus was just lacking activation of P65 or whether it actively suppressed P65. And so we did the following experiment. We took Zika virus infected DCs that were infected for 23 hours, and then we stimulated them with KLA, which is a very selective TLR4 agonist. And then we looked at gene expression 1 hour later. And of the 700 genes that are induced by KLA in a typical DC, we found that the vast majority were stimulated to a much higher degree in mock infected cells than in Zika virus positive cells. If the stimulation would have been the same in both populations, all the dots would cluster right around the central line. If they were higher in Zika virus positive, they'd be up here. If they're higher in Zika virus in mock, then they're down here. So Zika virus, in this case, is actively suppressing TLR4 mediated NF-kappa B upregulation of target genes. And so this seems to be an active process. And so the conclusions from this third part are that Zika virus suppresses NF-kappa B activity, and that impairs both DC and T cell activation. So Zika virus downregulates expression of core DC functional programs, and that includes downregulation of critical co-stimulatory molecules like CD40, CD80, CD86, critical cytokines for T cell production like IL-12, IL-27. It downregulates CCR7, which is a critical receptor important for DC migration. And it also downregulates cytokines that are really important for stimulation of bystander cells like IL-6, TNF, and IL-1 beta. Zika virus infected DCs do not mature or poor cytokine secretors and do not activate T cells. We just talked about that. And mechanistically, Zika virus inhibits NF-kappa B activation in DCs. So overall, the conclusions for this talk are that Zika virus is an important disease that I don't think any of us should forget about. I think that there is a great potential for this virus to cause a significant epidemics in the future, and that with devastating consequences. Two, that we've developed an integrated genomics method that can be applied to transcriptional assays like RNA-seq, genomic assays like ChIP-seq, csRNA-seq, ATAC-seq, all in virus to look at virus-specific responses. Zika virus and dengue virus both inhibit downstream type 1 interferon signaling, but the activation of type 1 interferon is delayed, and that allows initial infection of the infected cells. Zika virus stimulates specifically really no other pathways but SREBP-dependent lipid biosynthetic pathways, which is quite remarkable. For a cell that has 20% of its RNA, viral RNA, it only has like a very specific pathway that's really being stimulated. And this drives viral production. And Zika virus suppresses NF-kappa B activity and pro-inflammatory signaling, which disarms both innate and adaptive immune responses. With that, I'd like to really thank, this has been a highly collaborative effort, especially with the Shrestha lab. Sujin Shrestha was a mentor for me and now a very close collaborator. And all of these projects have been done in close collaboration with our lab. Emily Branch and Yingting Wang have been really critical in multiple of these stories. I'd like to thank the people in my lab who have been actively participating in this project. You can see them here. And then a lot of my UCSD collaborators, Christopher Glass, my mentor, Christopher Benner, Sasha, Nathan, and Sven are all active, close collaborators. And with that, that's it. And I want to thank my funders, Burroughs Wellcome and NIH and the Prepare Institute. I see Doug's hand up. If you'd like to ask a question, Doug.
Yeah, sure. Thanks. So that's really terrific work, Aaron. It was a very nice presentation. So this is not an area that I'm on top of, but I had a few thoughts. These are two viruses are so closely related genetically, but what you showed is the remarkably different activation and transcription responses. And dengue has been, I don't know how long it's been circulating in humans, millennia or whatever, and it's a human-human transmission. And if I'm not mistaken, Zika was isolated from a monkey about 40, 50 years ago in sub-Saharan Africa. And the question is, are those factors just coincidental, or could there be a difference in the evolutionary adaptation? And I'm curious if the original monkey isolate is like the other human isolates that you described in terms of the transcriptional responses. And if you can speculate how the different phenotypic behaviors could have evolved from such genetically similar agents.
Yeah, I'll try to answer those questions to the best of my ability, Doug. Thanks. Yeah, so is the fact that Zika virus emerged so recently, is that one of the reasons that we see different transcriptional responses? And it definitely could be. And I trying to think of exactly how to test that unless we have ancient isolates of dengue virus.
Are the NF-kappa B promoters or responses different in the monkey and the human in any way, or?
Oh, that's an interesting question. Yeah, I mean, potentially there could be differences in recognition or activation. You know, many of the inhibitory mechanisms of these viruses used to attack interferon are very species specific. So in the case of dengue and Zika, they can't inhibit, you know, it can't degrade stats in mice, you know, in mouse cells. And so they require IFNAR knockout or other types of interferon knockouts in order to infect them. That's not true for some of the other flaviviruses that have broader host tropism, like West Nile and some of these other viruses that have to infect birds as well. And so, you know, it's quite possible that some of the innate immune defense mechanisms that are present in these viruses are going to be different depending on the species that you're in. So I think that that's a really good question. And I can't answer definitively, but I think that's a really interesting thought. You know, oftentimes we think of viruses that newly emerge into a host, oftentimes tend to be more pathogenic, unless, you know, correct me if I'm wrong. I think about viruses, we think that they may host adapt over time and become less kind of inflammatory, less pathogenic, and better at transmission. But these viruses also have to contend with the fact that they have to move back and forth between mosquitoes and different animals. And so maybe that restricts some of that coevolution, I guess. Yeah, I don't know. A lot of speculation. I'm not sure if I can answer that specifically.
Okay,
Aaron,
an answer, let me know.
Yeah, I'll keep thinking about it. Oh, about the original monkey isolate. Yes. So the Dakar strain is an African lineage, and it's an old virus. I can't say exactly when that was isolated, but it was isolated decades and decades ago, and it seemed to have the same response. And so it doesn't seem like there's been a huge amount of evolution of this response over the past, you know, 30 years or so.
Sinem, I think you were next, right?
yeah, great job, Aaron. Thank you. Very interesting talk. SRABP, I may be wrong about this, but is SRABP also involved in iron metabolism, or is it the wrong SRABP I'm thinking about? I think there are multiples of those, but I don't know which one would be in the iron metabolism.
It may be involved in iron metabolism. I'm not aware of it. I usually, you know, they're usually considered kind of master regulators of lipid transcription. However, they may be involved in other gene synthesis pathways.
Is there any sort of clinical manifestation related to iron levels in the patients?
Good question. Not that I know about. I definitely could have missed this. I don't know of individuals with iron overload syndromes who would be more susceptible to disease than others.
Mm-hmm.
But I could just, you know, that could be present. I just don't know about it. I see Mary Garette's hand.
Yes, nice talk, Aaron. And we miss you here, by the way. Anyway, so this is a vector-borne disease, and my background being entomology, I'm interested in how do we know how many mosquitoes are actually infected with this virus? Because I can imagine not all mosquitoes are. Do we know the percentage of mosquito population that are infected with this? Because not everybody who gets a bite gets bitten by the mosquito is not gonna get the disease. Do we know the percentage?
It's a good question. I imagine that's highly dependent on, you know, the location and the, you know, extent of an outbreak, and maybe even the rainy season and other things like that may play into that. You know, during I can't even give you a range, unfortunately. I wish I could give you a percent range in certain populations, but you're right. I mean, there are probably plenty of cases in which a bite, even with an infected mosquito, did not end up in causing an infection in a person. I think there's probably a lot of factors that go into that. These viruses, the Aedes mosquitoes they've just been so successful at adapting to urban environments and living with humans and basically biting humans repetitively. And so I think that just makes them a very formidable mosquito host for this virus. It's similar to the way that we're starting to see malaria move into different Anopheles populations that are better at cohabitating with humans, how that's just really a scary scenario because it just increases the risk of transmission in areas that potentially were not as highly susceptible to transmission as previously. And so this mosquito is just really a big issue.
Thanks.
Yep. I see John's hand.
Hey, Aaron, great talk. Really
Thanks.
fantastic. What, maybe I missed it though. Do clinically available, cholesterol lowering drugs have any impact in these replication assays?
Yeah, so we did try some like statin drugs and stuff like that because these are so widely utilized in the population, right? And people speculate a lot about whether statin use in COVID or statin use in other types of infectious diseases has had clinical benefit. We know from decades of research that in individuals who have systemic viral infections or are treated with type 1 interferons like we used to do for hepatitis C treatment, right? That would lower cholesterol levels. So this is part of a normal immune response to viral infection is to suppress the production of cholesterol and other fatty acids. Part of that may be through the production of CH25H, an interferon-stimulated gene that makes 25-hydroxycholesterol, which a modification of cholesterol that can feed back and block, which is similar to DMHCA, the production of the activation of SREBPs by trapping it in the ER. And so I think that this is a typical host defense, but it really requires the interferons themselves to work, right? Like basically the host has to have seen the interferon in order to get that response. I think that so in our assay, statins had an effect. It was not as significant as DMHCA. And I have a couple of hypotheses for why that might be the case. So statins target HMG-CoA reductase, which is the rate limiting step of cholesterol metabolism. But so what happens in that scenario is you suppress cholesterol production. You basically suppress it at the early rate limiting step, right? But what that does is it activates SREBPs because there's no feedback. And so now you start producing all the other enzymes in the pathway at high levels. And so if you're able to scavenge any intermediates, you could still produce end product cholesterol at high rates, even in the presence of a statin. Whereas, with an SREBP antagonist, you are inhibiting basically the entire pathway. It can't compensate for that because there's no feedback anymore to the transcription factors that are responsible for activating the pathway. And so that may be one of the reasons that we see an even more pronounced effect with the SREBP antagonists. There may be other effects too that we're also interested in. There's a lot of crosstalk between SREBPs and lipid metabolism and interferon and inflammatory responses. And so we're very interested in how activation of SREBPs themselves also feed back and change inflammatory signaling and vice versa.
Thank you.
Yeah.
All right, we're at 2 o'clock, so I'm just going to quickly share the slide for our next month's presentation. All right, here. So next month we're scheduled to have Drs. Rednesh Lal and Sandra Leibel, who are recipients of our Prepare and HMI Collaborative Development Grant. So we'll get details as far as exactly when that'll be, and we'll be sending out those notices in the coming weeks. But thank you all for attending. Aaron, that was a tremendous talk. Thank you so much. Good news about your color choices. Looks like Davey and Mary both they could see them. So props to you again.
Great.
But Thank you all for attending our Prepare seminar series. We're extremely grateful for your interest and your participation, and we look forward to seeing you all in the coming year. Until then, have lovely holidays, and we'll chat soon. Take care.
Bye, everyone. Thanks.